So this is chapter 28, and I'm going to do 27 in the makeup lectures, because this one I think is probably more likely to be stuff you've never heard before, or in general. I'm going to spend, in invertebrates, we're going to talk a lot about organisms that you may or may not be at all familiar with, or that you might not know very much about. And so we'll start, like I said, we can't possibly talk about even all of the phyla of invertebrates. We keep discovering new phyla. And we certainly can't talk about all of the classes, let alone any unique species. So I'm going to just be waving my hands over invertebrates and giving you little highlights of generalizations. Okay. Invertebrate zoology is easily a full semester by itself, just looking at the different phylas of invertebrates. So we're going to have to hit the highlights. We'll start with these independent phyla, Porifera and Cnidaria, and then we'll talk about these superphyla and hit some of the key groups in each of those three superphyla. So this is just here for a reference point again. We're going to start over here with our Porifera, the sponges, and you'll notice that the Porifera do not have tissues. So they're not diploblastic, they're not triploblastic, they're on their own because they don't have any tissues. Okay. I have this picture just to give you some idea of the different shapes that sponges can take on. They can be very small, like these boring sponges are actually small, superficial, creating almost a crust-like surface over that coral skeleton, up to barrel sponges that can be, a couple of feet in diameter and a couple of feet tall, these huge structures. Okay, they really vary in size depending on what type of sponge they are, and they vary greatly in their shapes. Some of them are like a big, almost solid lump, like a bath sponge. They can be fairly large in size. Others have these tubes or columns. They look like fingers. They have these elegant shapes. So they really have a lot of different morphologies that they can take. They also can be lots of different colors. So they belong to the phylum Porifera. I am generally going to give you a Latin name and a common name when both exist. And I will probably also try to make sure that both of them are on any exam question I write. Okay. I would rather that you have a general idea of what the organism is like and worry less that you memorize a bunch of foreign Latin names for those groups. This one is kind of easy. Porifera, pores. The sponges are the hole bearers. That's what their name means. So they're full of pores, canals, and chambers. They're the simplest animals. They have no true tissues. So they are at the cellular level of organization. Their cells function as a group and share resources with each other, but they don't actually form a true tissue that has a single goal. They have different cell types that are all kind of like embedded in a medium together in a collection. They don't have any body symmetry. Some of them, barrel sponge, might look like they're perfectly circular and that they could have a radial symmetry, but they actually don't, especially if you cut them open and start looking at the chambers and the canals. Those are very asymmetrical across size. They can be really amorphous in their overall shape. Because they don't have true tissues, they can't have a body cavity, right? So they're not even acoelomate, they just don't have a body cavity. They don't have tissues to form a space. They're predominantly marine. The vast majority of species are marine, although there are a few freshwater sponges, and there's some estuary sponges that live in those transition zones between marine and freshwater environments. They're probably most abundant in reef systems. You find them in coral reefs. There are also some in some tide pools and rocky shores. As adults, they're sessile, which means that they can't move. They are anchored by a hold-fast structure at the base of the sponge that attaches to a substrate. Okay. When they are larvae, they are free swimming. And so in their juvenile state, they are mobile and can travel to a new location. But once they settle down and become an adult, they're going to be locked into that position unless in rare cases, if you're a small sponge, you might get picked up by something like a decorator crab. And so you can actually break off a piece of sponge, like if a certain species of sponge get damaged in tide action or in a storm, the broken pieces can kind of roll away and become new sponges. And crabs can actually pick them up or pinch them off of their parent and put them on their back and then carry them around. Decorator crabs like to put all sorts of things on their shells, and in that way the sponge gets to travel and go to other locations. Each sponge is a hermaphrodite. They have both male gamete, or both gametes are produced within the same species. I don't know why I'm having a hard time saying what I mean to say there. They produce both sperm and egg, which they release into the water, and fertilization is external in the water. Okay, this is the basic body plan of a sponge. And so we're looking, if you're looking at those finger or tube sponges, this would be one of the tubes, or the entirety of a barrel sponge. They have this same basic structure. And so the body of the sponge, what you see, is mostly made up of this substance that we call mesohyl, which is sort of a semi-fluid matrixy substance that holds all the cells together. And then they have several unique types of cells. And so over here we have the choanocyte, or the collar cell. In yellow, we have the spicules, and those are usually made of either silica or calcium carbonate. And so they give the sponge a firm, rigid structure within its body. Also make it pretty unpleasant to eat. There's not a lot of things that really eat sponges. They have these pore cells, which are called dermal ostia, and they pass through the outer surface of the sponge and into the center. And the center region is the main chamber, and the opening to that chamber is called the osculum. And so water will pass through the dermal ostia, in through the pores, and come into the central chamber. And then we also have, in blue here, amoebocytes. And the amoebocytes in the sponge are essentially like stem cells. They are sort of originate cells that can differentiate into other type of cells and also carry out basic metabolic function for the individual. So the this is what I want. The individual collar cells, or the choanocytes, are the feeding cells, and they are embedded all along the inside of that chamber with their flagella pointing into the open space where the water is flowing through. Their flagella are going to beat constantly, creating a water current, and that water current is going to draw fluid in through the pores into the center. So here are all these choanocytes with their flagella pointing inward. They're beating their flagella and creating a current. So water comes in and then goes out through the central osculum. As that water passes through, material in that water is going to travel across the collar cell, and food particles are going to get trapped on the surface of the collar. Okay? And those food particles will stick to the collar, and there's a little bit of a fluid-like substance over the surface of the collar. And so the food particles will move down the collar towards the surface of the cell, where a food vacuole can form and come into the cell for digestion. Okay. So these are the feeders of the cell. They're going to be feeding the rest of the sponge. Oops, wrong way. The outer cells, shown here in blue, are the epithelial cells. They form the outer protective layer and hold that mesohyl substrate in place. The pore cells, again, have lots of names, the dermal ostia. I am not going to test you on these different cell types in the sponges. The pore cells create the opening for the water to come in. The amoeboid cells produce the matrix, and they produce the spicules. The collar cells do the eating, and the spicules provide structural support. Okay, so they have all these unique cell types that are carrying out all the functions of life, but not necessarily forming true tissues. Okay, do you guys have any questions about sponges? They're pretty simple. All right, so we'll go ahead and talk about the cnidarians. The C is silent, pronounced cnidaria. And this includes a pretty diverse group of organisms, all of which possess cnidocytes. And so the name cnidaria comes from these specialized cells that they all contain. So they range from sessile organisms like the hydra and anemone and coral that have this polyp shape that are attached to a substrate, to a whole diversity of things that we think of as jellyfish, which have a free swimming shape. The Portuguese man-o-war that is shown here is not actually a single individual, but is instead a mixed colony containing both polyp forms and jellyfish forms, medusa forms, in the same colony. And there's more than one of these sort of colonial living organisms in the cnidarians that are just really fascinating. All coral are cnidaria. Porifera sometimes grow on the coral. So sponges will sometimes grow on a coral reef, and that crusting sponge or that boring sponge that was in that picture was actually growing on the dead skeleton of a coral. So is the sponge growing over the coral as opposed to being the coral. Sponges are frequently an important part of the coral reef ecosystem, and they live on the skeleton. So the coral reef, if you think of just looking at a section of reef, that reef is the dead bodies of all of the generations of corals that have lived in that space. And new layers build up on the old layers. And we have this ability to grow that reef. The live sections are the ones where we still have those individual sedentary organisms attached to the corals. Okay, so cnidaria are categorized by the presence of cnidocytes, and these cnidocytes are singing cells. We'll look at those in a minute. They actually have tissues, so they move up one step in their complexity. They have true tissues, but they do not have any organs or organ systems. Okay, they are radially symmetrical, so their body parts reflect around a center point and are evenly distributed around that point. They have what's known as a gastrovascular cavity. And so we're going to use that term in many of our organisms. The gastrovascular cavity is the digestive system. In many cases, it is also the circulatory system and the respiratory system. It's responsible for delivering nutrients and responsible for delivering gases. And this gastrovascular cavity has a single opening. And so their mouth is also their anus. Food goes in, water goes in, oxygen goes in, waste gases go back out the same opening. They can close and open that opening between those two processes. They don't have a body cavity. They have simple contractile tissues and a simple nerve net. They don't really have a nervous system, but they have nerves that are connected over the entire surface of the animal's body, forming what we call a nerve net. They're predominantly marine. There are some freshwater species, but you don't ever see them because they're really tiny and they're on the surface of rocks, and they're mostly transparent in appearance. Most cnidarians, at some point in time in their life, have both body morphs, the polyp and the hydra. So they're polymorphic. I'm sorry, the polyp and the medusa. Polyp and hydra are the same thing. So the polyp shape looks like a hydra, the medusa shape looks like a jellyfish. And most species, all species, have both of those forms at some point in their life cycle. So we'll stop there with Nightarians for our regular class time. If you want to stick around for the double makeup lecture, I'm going to actually talk about chapter 27, homeostasis. We'll continue with chapter 28 on Thursday. If you are not going to Harvard, then don't come to this class on Tuesday because we'll all be in Cambridge. Please make sure you've let me know that you're not going. And if your plans change, you wake up Tuesday morning, you're like, I can't go, please tell me so that we're not all sitting around waiting for you with the van. I think it says 10. So we're going to leave at 10 from the space in front of GCC where the buses drop people off and pick people up, that little bus stop. We'll meet there at 10. If you're driving your own car, it's probably easiest to meet us there and sort of follow us down Route 2, unless you're familiar enough with Cambridge that you know where you're going to park. I will pay for parking passes in the Oxford Street Garage. Trying to feed meters is really not worth doing. And if you're not parking in the Oxford Street Garage, if you're driving yourself, you can park at Alewife and take the MTA in. We go right past the Alewife station, if that's your preference, whatever you want to do. Yeah. I'll start at 3.